Hey everyone, welcome to another week of Ecology Lab. This is Erin, and I'm going to take you through the second part of our population ecology lecture, which is focusing on population growth. So, as we talked about last week, and actually the week before, a population is a group of individuals that are interacting and of one species that occupy a defined geographical area. So, often when we're talking about whatever our study subject is, we're talking about a population of a species within a specific area. So, when we talk about the concept of population growth, we also need to define what we're talking about when we talk about organisms. So, when we talk about unitary organisms, these are kind of easy to identify as individuals that have clear boundaries, as in like within a certain species. So, we're talking about like all humans or all palm trees or dogs or whatnot. A modular organism is a little bit more ambiguous as to where you separate one individual from another. So when we talk about this, we're talking about maybe genetic individuals or individuals within like a colony, like clonal colony. So, for example, when we talk about like coral reefs, like we have one coral head and it's like multiple coral polyps that are all the same genetically. So it's like a little bit more unclear that is like one unitary organism, right? So we are talking about multiple individuals that maybe have the same genetic makeup or whatnot. But we're really going to be focusing, fortunately, because it's more simple, on unitary organisms. So ones that are really clearly identified as like individual organisms. So one example that we're going to go through today is looking at this type of tree, the trembling giant, Populus tremuloides, which is one of the largest known organisms on the planet. Our study system is now looking at the Fish Lake National Forest in Utah, which is about over 100 acres, and collectively all of these trees weigh 6,600 short tons. Their root system is an estimated 80,000 years old. So when we're talking about this whole system, we're talking about like the features of this population. We're focusing on size, which is the number of individuals within an area, density, the number of individuals per unit of area or unit volume. So an easy way to think about density is like how close together are the individuals, right? So you could have the same size, 100 individuals or 100 individuals, but their density could be wildly different. So 100 individuals packed in a smaller area compared to 100 individuals kind of spread out over a much larger unit area. Dispersion is the spacing of organisms relative to each other. So, for example, we could have some organisms that are tightly clumped together, so like low dispersion, or really widely spaced out. So, this goes back to a little bit talking about our sampling methods. So, depending on kind of what we want to study, it'll mean a different technique and a different method when it comes to kind of quantifying our entire population. So to start at the top, like total counts, this means that you're counting literally everything in the population, which, as we talked about, is great because it is the most accurate way of getting the information about your population. However, we often don't do this because the disadvantages are great in the sense that it's mostly impossible as far as the time it takes to count all the individuals or measure all the individuals, and also the resources it takes. So disadvantages, it's often too difficult, too expensive. So quadrats is another sampling technique that we talked about a couple weeks ago, where you've got your kind of defined unit of area and you use a randomized technique to kind of quantify whatever falls within your quadrat. So the advantage is that it's pretty easy and cheap to do, and it can be really effective for things that don't move, like plants, for example. Disadvantage, it has to estimate the entire sampling area, so you have to make sure that you're getting enough of the different types of habitats. Sorry, shutting the door, it's very loud. You have the different types of habitats to represent the whole area. Also, there's opportunities for error, although this can apply to multiple different types of sampling methods. One that we didn't talk about as much the capture, recapture, or mark recapture. You capture all the organisms in a known area, mark them, release them, let them disperse, and then recapture them. So this is a way that we can see where organisms go, or how many stay within a certain area or move outside a certain area. This is useful for large populations and good for highly mobile species. This is something that we do with sharks, for example. However, marking can be invasive. It assumes movement or that the organisms is random, and then it also requires that the individuals need to disperse within the area. One that we talked about again when we did the plant cover was percent cover, where you estimate the percentage of coverage of a patch of a known size. This can be really good for species that don't move or that are easily defined, although as we know, it can be inaccurate and have high experimental error because it really kind of depends on kind of a personal assessment of what you think the percent cover is, which can really vary from person to person. So again, these are 4 ways that we can define population density or measure population density. And depending on the species that you're using, whether it's a mobile species or a really sedentary species, it can kind of affect which technique that you use. So there's really 4 main parameters that influence population density. And one is birth, how many new individuals are born into a population. Death, how many individuals are removed from the population from natural mortality. Immigration, the number of individuals that move into a new area. And emigration, that the number of individuals that move out of an area, right? So when we think about population density is the number of individuals that occupy a specific unit of area. So thinking about how many are born, how many die, how many come in, and how many leave from that area. So when we have that information, we can calculate the population size. So here's the formula for population size. We have N sub T, and so T here represents time. So the number of individuals or the population size at time T equals N sub 0, which is the population at time 0. So what's our starting population, right? Plus births minus deaths plus immigration minus emigration. So this might seem a little bit complicated, but if you break it down, it's pretty logical, right? So like the number of individuals that we have at whatever time we're interested in is equal to the number of individuals we had when we started, plus the number that were born and that moved into the area, minus the number that died and moved out of the area. So again, N sub T, the number that we have at the start, plus births, minus deaths, plus immigration, minus emigration. So to model population growth over time, we need to know a couple things. First, we need to know about the generation types. So do they overlap or do they not overlap? So when we think about generation types overlapping, like one overlapping is really clear, which is people, right? So we, the offspring, overlap with our parents' generation that overlap with their parents' generation. So there are periods of time where all of us exist at the same time, right? But a non-overlapping species would be like, when the parents give birth to the offspring and instantly die, or they die when they lay eggs, so that the offspring never actually are alive at the same time as the parents. So an example, of course, elephants, overlapping generation, very similar kind of population structure to humans. And then non-overlapping generation, a lot of insects or some fish where you lay your eggs and then instantly die. Another example is the reproductive type, which is continuous or discrete. So when we talk about continuous generations, that means that like offspring can be born at any time, right? Like people are this way. You can have kids at any time of the year. It doesn't depend on the seasons. People can just like reproduce whenever they want. But discrete generations are like, the parent generation needs to get to a certain age and then they all reproduce at the same time and then they all die at the same time, right? So like the generations are more of this stair step. You can see like the blue steps to the right. Those are discrete generations. Continuous is just like there's an increase in offspring kind of like steadily throughout time, as opposed to like in pretty distinct time segments where like the offspring are born. And the other thing is you talk about simulpareous, which means you only reproduce one time, versus iteropareous, which is reproduce multiple times. So there are some species, for example, salmon, they die after spawning. So they reach maturity, they spawn once, and then they use all their energy to spawn their offspring so that they die. So they only have the opportunity to spawn, to give birth one time, reproduce one time. But then iteropareous, and like many mammals, including people, humans, that like you can reproduce multiple times, which has, of course, a great effect on kind of the distribution of offspring over time. So the other thing to keep in mind is that populations do not just grow. Like if you think about it, you're like, well, of course, like if these populations grow to a certain size and everyone's giving birth, then like maybe we just have like exponential growth, right? And when you think about like, how often can you give birth and how they die and discrete versus continuous and types of growth, the thing that really comes up is like, ultimately, you will hit a wall when it comes to resources. So there's only, like in theory, yes, you could just continue giving birth. A population could just keep growing and growing and growing. But ultimately, they're going to hit confines in the number of individuals that habitat that they live in can sustain. And we talk about that in terms of resource limitation, which a simpler way of saying that is like there's only so much food that can go around, or so much nutrients that can go around, or space, or mates, or whatever it is, right? So a lot of times in ecology, when we talk about like, how organisms live within their ecosystem, we are really, really interested in what are the resource limitations within that habitat that like would ultimately kind of control the population growth of those animals, right? So some of the things that we think about when it comes to resource limitations is things that happen over time. So we'll get into this a little bit, but sometimes there are like natural events that will naturally curb the population growth. And then other things, we talk about competition, predation, herbivory, and disease. So these are effects from other organisms or other maybe parasites in the environment that would ultimately curb the ability of these populations to grow unchecked, right? To grow indefinitely. So when we talk about things growing indefinitely, like with no resource limitation, we talk about that in terms of geometric growth. So that's the red line down here on this chart, where the model of population growth is just assuming that the resources are not limited. So you can see geometric growth is also the same as exponential growth. Oh, you see time is on the x-axis, and over time this population is growing, 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 and then you get to a certain point and that it just like skyrockets, right? Because there's just so many organisms within the population reproducing that it increases exponentially. This is unrealistic in nature for long periods of time because, as we know, resources will ultimately limit the ability of that population to grow. And also think of it, I mean, we're seeing it with people, right? Like we talk about the sort of resource limitations of our planet and how many people this planet can sustain as far as like physical space and also food availability and that kind of thing. This can occur on small timescales. So if we're looking at kind of short periods of time, you might be able to see this sort of exponential growth. After disturbance or species invasions, which is another way of saying like essentially something disrupts the kind of natural structure of the environment that would allow one species to kind of take advantage of that situation and just like skyrocket their population. But mostly what we talk about is logistic growth, which is this blue line in the model next to us, which is a model that assumes that resources are limited. So you can see both of the geometric and logistic growth curves start in a similar way. They start at time 0 and they start to increase and they're growing and growing and growing. And you can see that like just around the time that geometric growth starts to take off, we still see an increase in growth in the logistic growth curve in that blue curve. You can see right around the middle here, if you can see my cursor, is the fastest rate of growth. The slope is the greatest right here. But then as it starts to grow, you can see how the resources begin to limit how fast that can grow, and it starts to kind of flatten out, right? So unlike this geometric growth that kind of is just to the moon, the logistic growth eventually starts to hit a wall with resource limitations and starts to kind of flatten out around this one population size, around K, which we're going to talk about. So K is a abbreviation for carrying capacity. So carrying capacity is based on this idea that habitats have a limited number of resources, and eventually this population is going to grow in such a way that it's going to hit the kind of maximum number of individuals that can appropriately share those resources within the habitat. The carrying capacity can change based on how resources change. So if you think about and maybe a tundra area where in the summertime there's grasses available, but in the wintertime there aren't, we might see like an increased number of individuals who feed on grasses that can be there in the summer than in the wintertime. But the official definition, carrying capacity, also known as K, the number of individuals that can be supported in a habitat at a given time. So again, this is the idea that like the habitat based on the limited number of resources can really only sustain a certain number of individuals before the resources are maxed out. Again, this can vary through time or space. This is not like a totally stagnant metric. The other thing is, this, what we just looked at, is pretty clean, right? The population increases over time. This is N, number of individuals, T, time. So the number of individuals increases over time. And it's this kind of really smooth loop up to carrying capacity where it just kind of settles out at carrying capacity. But that's not, of course, always what we see in nature. And a lot of times we do see fluctuations around this carrying capacity. So the relationship with the carrying capacity could change depending on our starting conditions. So we can see, stable is there's no oscillations, or kind of, if you think of like a curve, a sinusoidal curve, if you go all the way back to your calculus classes, like going around. So what we just looked at was a stable curve where there's no oscillations. So it essentially, the number of individuals increases to this kind of stable equilibrium, meaning there's not variation, and it just kind of stays along that carrying capacity number. We can also see convergent oscillations. So convergent, remember, comes together, converge. So what we're seeing is this super rapid rate of increase of the number of individuals. And then as you can see, we're growing, and we actually overshoot this dotted line, which is the carrying capacity, or K. So we overshoot K, which means there's more individuals than can be supported with the resources in the habitat. So if you think of, we're overextending the population, so that means the individuals are going to die because there is not enough resources to go around. So as these individuals die, you see this decline. You see this decline closer to carrying capacity, and then because this is like they really overshot it, a lot of individuals die, so we actually overshoot it again, but essentially undershoot it. And then this converging oscillation is this sort of fluctuation around the curve, but you can see the magnitude of each one of these oscillations decreases, and so we do hit that kind of equilibrium around K. Stable limit cycles is something that is close but a little different. So we're still fluctuating around K, but as you can see, we're not like way overshooting it and then way undershooting it. We're kind of keeping this steady, a little bit over and then a little bit under and then a little bit over, but like we kind of average around K. So the population will sometimes be a little bit too much for the habitat to support, and then some individuals will die and will kind of go down, but then there's more resources to go around, so then the population grows, and so on. Chaotic fluctuations is when there's not really a pattern. So we still see kind of over and under these sort of oscillations around carrying capacity, but like sometimes we'll see it way overshoot and sometimes it'll just be a little bit over. And so this can be affected by a lot of environmental factors, as well as some other random factors affecting population density. So density, when we talk about like what kind of causes those population numbers to go up or down and kind of oscillate around that carrying capacity, we talk about density dependent versus density independent factors, right? So density dependent, this you can remember because it's like depends on the density of individuals, or when biotic factors in the environment that have an increasing effect as population size increases. So think of it as like, all right, as we have more and more and more individuals, there might be a greater chance that disease spreads among the individuals, or there's more competition for resources, or it's easier to spread parasites, etc. So when we go back, when we look at these figures, like a density dependent factor, if we looked at this damped oscillation would be like we really increase numbers and then there's super increased competition for those resources, or because it's really dense, it's really easy for disease to spread, and that's what causes things to go down, right? So that is density dependent. These factors depend on increasing numbers of individuals. But density independent factors are different. These are abiotic, so not biotic, like not related to living organisms, factors that are in the environment that affect the entire population regardless of their density. So this can be temperature, weather, like if you have a storm event or a fire event, like that fire event is not dependent on how many individuals you have in that population. Or, we talk about some animals that maybe have great population growth in warm temperatures, but die off when it gets really cold. Like that temperature is going to get cold regardless of how many individuals there are in that population, right? So when we talk about density independent factors, those are abiotic factors that really, couldn't care less about, how many individuals there are in the population. So more examples of density independent factors. So when we talk about like some of these leaf eating insects like thrips or aphids, they can really dominate these plants and on their leaves, but then can be totally washed out by a heavy rainstorm. So that rain is coming no matter how many aphids are on a tree, right, or on a plant. But that's a density independent factor. Another one would be like waves that get really amplified by hurricanes can really cause big damage on coral reef ecosystems and wipe out certain coral reef colonies. And again, that storm is coming regardless of the density of the coral reef at that time. So another density independent factor. Also cold or hot weather, like severe drought or severe cold. Again, pretty much if you're thinking about independent versus dependent factors, like anytime you're talking about a like natural disaster or a big climate event, that's always going to be a density independent factor. So one of the really classic studies when it comes to density dependent factors is looking at Gauss in 1934. So he had these two paramecium species where he put them in the same system, one in the same system and one in different systems. So they're either growing together in the same kind of colony or they're growing separately. And so he tried to control all of these different limiting factors. So everyone got the same amount of food each day. All of them had their waste removed. All the temperature and other conditions were kept the same. And then he was trying to study the competition for shared resources. So down here, you can see that when they're grown separately, both of these types of paramecium, P. aurelia and P. caudatum, had the same kind of growth pattern, right? You see that sort of carrying capacity logistic growth where we increase and then we start to kind of level out around the same system. The same kind of carrying capacity. It's pretty similar. But then on the bottom, when they're grown together, P. aurelium still follows that pattern, but you can see P. caudatum kind of starts to increase and then really crashes to the point that there's like practically none in that system. So we can conclude that when they're grown together, the reason they have different growth rates is actually because of competition for shared resources, and that P. aurelia is actually a better competitor. So when it came time to like the population got large enough that they start to have to compete for resources, P. aurelia just like stomped P. caudatum. Like they just totally took over, were able to drive them essentially to extinction in this experiment. So again, another component of density-dependent rules, we have Yoda's law, or the negative 3/2 power rule. So as plant density rises, competition also rises, and then competition has several effects on plant biomass. So I know this figure is kind of funny. It's like not a super great figure, but you can see as on the x-axis, when the density of the plants increase, the mass of the plants decreases, right? So as plant density rises, there's more and more competition, which has all these effects on plant biomass. So there's fewer resources to go around per plant, so more competition for food and light, which are also important for growth and survival. And there's also physical changes due to limited space. So we see that, like, again, more and more plants that are there, it means that, like, for each individual plant, the mass that they can achieve is decreased. So this is a type of functional response. And this is an important concept in ecology, where it's the intake rate of a consumer as a function of food density. So this is the amount of food that an individual is able to take up as it relates to how much food is available. So there's a couple different types. So type 1, a linear increase in predator intake with prey density. So that means that, like, as there's more and more prey, predators are able to eat more and more food. This kind of assumes that there's no effort on the predator's side of having to search and kind of handle food. So when we talk about handling time, it's essentially like, how much work does it take for that predator to actually intake that food? So an example would be like, there's some, an otter, for example, likes to eat shellfish. So the otter is able to find these shellfish, but it comes with this, like, hard outer shell. So their handling time would be like the time between when the otter gets the item and has to break into that shell to actually eat the food. And handling time can take a lot of energy. It can also mean, depending on your organism, it could mean that, like, you're exposed to predators during that time. So when we talk about, an increase in predator intake related to prey density, it means that, like, we're assuming that there's no kind of additional effort of, like, having to find additional food or handle the food by the predator. So it's a really easy prey +1, predator +1. So type 2 is a decelerating predator intake rate, which means that, like, as the amount of prey increases, the amount of prey a predator can eat actually decreases. And that really assumes that, like, they essentially just don't have enough time to, like, process all the food, even though it's increasing. So handling time puts a limit on how much prey can be consumed. So as prey increases, it might actually take more time for them to be able to handle that prey to actually eat it. Type 3 would be a sigmoidal or S-shaped predator intake rate. So that means that, like, it is low intake rate at low prey abundance, which could mean that it's hard to find food, for example, a high intake rate at intermediate abundance, and then a low intake rate at high abundance. So think of it as, like, kind of a parabola, right? So, or S-shaped when we talk about, like, the logistic growth curve, where at, like, low levels, there's low amount of consumption. At high levels, there's low amount of consumption. And then at intermediate levels, it's, like, just right. We're a little bit of a Goldilocks situation here, that, like, there's just enough prey that the search time is low, but not too much prey that the handling time is too high. This is also often justified by encounter rate. Again, that's search time, learning time, prey switching. So here is an example of, like, when we look at on the x-axis, as the density of prey increases, so there's more and more prey available, we have these different sort of functional responses, right? Like a 1, 2, and 3. So if we look at this right here, the type of 1 is like, okay, as prey density increases, we can see it's pretty linear, that the number of prey consumed by a predator just skyrockets, right? It's a super clear linear relationship. Type 2 would be that, like, as we increase, you're seeing an increased rate of prey consumption, but then it kind of levels out over this like upper level of prey density because of increased handling time, right? So let me just jump back. This is this asymptotic predator intake rate. So essentially, handling time will ultimately put a limit on how much prey can be consumed. So once it increases, and then ultimately The handling time will mean that like you just can't take in any more prey. And then type 3, you can see it's kind of low rates at low numbers. It starts to really increase at this intermediate prey density. And then you can see the rate really decreases again at that high prey density. And then the same idea, but a different way to conceptualize it. When you see on the y-axis here, proportion of prey consumed by predator, type 1 means that like no matter what the density of the prey population, the proportion of the prey consumed by each predator will remain the same. For type 2, you can see the predation rate decreases as you kind of get to this upper limit of food consumption. And then in type 3, the predation rate really decreases at low levels and at high prey densities, but it's the highest proportion of prey consumed by predators at one of these intermediate ranges. So, excuse me. The Holling disk equation, developed by C.S. Hollings, describes the relationship between search time, how long it takes to find the prey, handling time, how long it takes to actually be able to consume it after you find it, and then consumption rate, which is actually eating it and digesting it. This was developed by running a simulation using blindfolded humans and small discs of sandpaper on a flat surface. So essentially, how fast people can find and consume things like based on what's available. So we're actually going to do our own little simulation today of the type 1 and type 2 functional responses to see if we kind of find something similar. We're interested in a couple parameters here. N would be the density of prey, so like how much prey is there per unit of area. A would be the attack rate or searching efficiency of the predator. So how frequently are you able to capture the prey, right? The T subtote is the total time spent foraging or looking for prey. S is the total search time for all prey. T sub H is the handling time per prey item. And P sub E is the number of prey eaten during the period of searching. So I'm actually going to stop there and let Drew explain this system, the activity that we're going to do today. But you can find the instructions on the PowerPoint that is uploaded on our module for this week. And if you have any questions, ask him. He is the other awesome TA for ecology today. And then if you have any questions about this lecture, you can either ask him or feel free to email me, and I'm happy to answer any of your questions. But yeah, otherwise, enjoy today, and looking forward to diving into this next time I see you. Thanks so much.